Every year, millions of pregnant women take medications for conditions like depression, epilepsy, high blood pressure, diabetes, and infections. Yet the vast majority of drugs available today lack adequate safety data in pregnancy. This is the pregnancy paradox. We have been so afraid of studying drugs in pregnancy that pregnant women are routinely treated with drugs that have never been properly studied in their unique physiology. How did we get here, and what can we do about it? The scale of the problem is staggering. Over 90% of pregnant women take at least one medication during their pregnancy. The average is 3 to 5 different drugs. Yet fewer than 1% of approved drugs have adequate pregnancy safety data from clinical trials. More than 80% of medication use during pregnancy is off-label, meaning it has never been studied for safety and appropriate dosing in pregnant populations. The same drugs that are carefully studied in clinical trials for the general population are prescribed to pregnant women without any pregnancy-specific evidence. The exclusion of pregnant women from clinical trials has two roots. The historical root is the thalidomide tragedy of the 1950s and 1960s, where a morning sickness drug caused over 10,000 severe birth defects. This was followed by the DES disaster, where a drug given to prevent miscarriage caused cancer in the daughters of treated women decades later. These tragedies led to a protective reflex that effectively excluded pregnant women from drug research. The contemporary barriers include fear of liability, the physiological complexity of pregnancy, ethical concerns about fetal risk, and cautious institutional review boards. Well-intentioned protection has created an evidence vacuum. Exclusion sounds protective, but it creates its own problems. First, when pregnant patients need medication, doctors prescribe based on animal data and expert opinion, not human trial data. This is the opposite of evidence-based medicine. Second, some pregnant women avoid necessary medications because of unknown risks, leading to untreated depression, uncontrolled epilepsy, or poorly managed hypertension, conditions that themselves harm both mother and baby. Third, pregnancy changes how the body processes drugs. Blood volume increases by 50%, kidney filtration increases by 50%, and liver enzyme activity changes dramatically. Without pregnancy-specific dosing studies, we do not know the right dose. In 2015, the FDA replaced the old pregnancy letter categories with a new system called the Pregnancy and Lactation Labeling Rule. The old letter system had been used since 1979 but was widely misunderstood. A drug labeled Category B, no evidence of risk, often meant no human studies had been done at all, not that the drug was proven safe. The new PLLR requires narrative summaries that include data from pregnancy exposure registries, risk summaries, clinical considerations and dose adjustments during pregnancy, and information about labor and delivery. Importantly, the new rule requires labeling to be updated as new data becomes available. Pregnancy fundamentally changes drug pharmacokinetics. Blood plasma volume increases by about 50%, diluting drug concentrations and often requiring higher loading doses. Kidney filtration rate increases by 50%, drugs cleared by the kidneys are eliminated much faster, so their doses must be increased. Liver enzyme activity changes dramatically. CYP3A4, which metabolizes about 50% of all drugs, is induced during pregnancy. Albumin levels decrease, meaning drugs that are highly protein-bound have more free active drug available. These changes can reduce drug concentrations at the target site by 30 to 60%. Without pregnancy-specific PK data, we are essentially guessing at the right dose. The debate between exclusion and inclusion is fundamentally an ethical argument on both sides. Arguments for exclusion include fetal vulnerability during critical developmental windows, the fetus cannot consent, the risk of teratogenic effects that animal studies may not predict, and the shadow of pharmaceutical disasters like thalidomide. Arguments for inclusion include the evidence gap that harms everyone, the recognition that pregnant patients can make informed decisions, the ethical obligation to study drugs that millions of pregnant women take, and the concept of dignity of risk. That excluding pregnant women from potentially beneficial research is itself an ethical problem. U.S. regulations already provide ethical frameworks for including pregnant women in research. The minimal risk standard allows research that presents no more than minimal risk to the fetus, such as a single extra blood draw for PK analysis. Many useful pregnancy PK studies could be conducted under this standard. When a pregnant woman has a medical condition, she can enroll in treatment trials that offer direct benefit, such as an HIV medication trial for a pregnant woman with HIV. The Task Force on Research in Pregnancy and Lactation, or PRGLAC, made 15 detailed recommendations to the NIH and FDA in 2018 and 2020 that address ethical frameworks, trial design, and regulatory incentives. Most pregnancy safety information comes from pregnancy exposure registries, observational studies that follow women who take specific medications during pregnancy and compare their outcomes to a control group. The Antiretroviral Pregnancy Registry has followed over 20,000 pregnancies and provides essential safety data for HIV medications. But registries have significant limitations. They can only detect relatively large increases in birth defects. They cannot determine appropriate dosing. And crucially, they only start collecting data after the drug is already on the market and thousands of pregnant women have already been exposed. Registries are necessary but not sufficient. Several innovative trial designs can advance pregnancy research while minimizing risk. Adaptive pharmacokinetic studies use sparse blood sampling, just 2 to 3 samples per patient, in pregnant women who are already receiving the drug clinically. These studies provide essential dosing data with minimal additional risk. Physiologically based pharmacokinetic modeling, or PBPK, uses computer simulations to predict drug behavior in pregnancy, and when combined with a small amount of real PK data, can provide robust dosing recommendations. And staggered enrollment starts with the third trimester, progresses to the second, then the first, building evidence step by step. The cost of our current ignorance is enormous. 6 million pregnant women in the United States take prescription medications each year, most without adequate pregnancy data. Approximately 30% of pregnant women with depression stop taking their antidepressants during pregnancy due to unknown risk, and their relapse rate increases 5-fold. Women with epilepsy face a similar dilemma. Uncontrolled seizures during pregnancy harm both mother and baby, yet their medication doses are often guessed rather than optimized. The medical costs from medication errors in pregnancy are estimated at over $4 billion annually in the United States alone. Several developments point toward a better future. Regulatory incentives modeled after the pediatric exclusivity program, where studying a drug in pregnancy would earn extended market exclusivity, could transform the economic calculus for sponsors. Decentralized trial designs are ideal for pregnancy studies since they reduce the burden of clinic visits on pregnant patients. And a growing movement toward default inclusion, where pregnant women are included in clinical research unless there is a specific scientific or safety reason for exclusion, is gaining support from regulators, ethicists, and patient advocates. Real change requires action across 4 domains. Regulation, implement the 15 PRGLAC recommendations and create FDA pregnancy study incentives. Institutional review boards, educate ethics committees that exclusion can be more harmful than inclusion. Pharmaceutical sponsors, include pregnancy PK substudies in Phase 3 development programs and pursue pregnancy indications. And patients, engage pregnant women as research partners. Many pregnant women want to participate in research that could help them and others. Their voices must be included in trial design and ethical review. So here is the bottom line. The reflex to exclude pregnant women from clinical research came from a noble desire to protect. But after 60 years, we must recognize that exclusion has created its own harms. The path forward is not reckless inclusion, but thoughtful, ethical research that builds evidence gradually, starting with minimal risk PK studies, using innovative designs, and respecting the autonomy of pregnant patients to make informed decisions about their own healthcare and their baby's well-being. Protecting pregnant patients means studying them, not excluding them.